Okay, Dr. Neelapu, can you give an overview of the study design of the phase 2 ZUMA12 study and highlight the updated findings?
Sure. So this is a phase 2 multicenter study of axi-cel as part of first-line therapy in patients with high-risk large B-cell lymphoma. So at this meeting, we are presenting the interim analysis from this study. So axi-cel was previously approved for relapsed or refractory large B-cell lymphoma after 2 or more lines of therapy. In this study, we decided to evaluate that as part of first-line therapy. To be eligible for this study, the patients had to meet at least 2 different high-risk criteria. At initial diagnosis, they need to have a double hit diagnosis of double hit or triple hit lymphoma or large B-cell lymphoma with IPS score of 3 or higher. In addition, we also had a dynamic risk assessment for these patients after they underwent 2 cycles of standard frontline chemotherapy with R-CHOP or EPOC chemotherapy. They had to have a positive PET scan, interim PET scan after 2 cycles defined with a dual score of 4 or 5. So once they are enrolled on the study, they underwent leukapheresis followed by conditioning chemotherapy with cyclophosphamide and fludarabine for 3 days, and then a single infusion of axi-cel product at a dose of 2 million CAR-positive cells per kilogram body weight. The primary endpoint for the study was complete response rate. At the time of interim analysis with the data cutoff of August 25th, 2020, 32 patients received conditioning chemotherapy and also received axi-cel CAR-T product. And we found an overall response rate of 85% and a complete response rate of 74% with a median follow-up of 9.3 months. 70% of these patients had ongoing response. And in fact, the median median duration of response, PFS, or overall survival has not been reached to date. The therapy itself has been fairly well tolerated. As expected, all patients developed cytokine release syndrome, but these were primarily grade 1 and grade 2. With 9% of the patients having grade 3 or higher CRS, and neurological toxicity was observed in 69% of the patients, but that, again, most of these were grade 1 and grade 2, with 25% of the patients having grade 3 or higher neurological toxicity. There were no grade 5 CRS or neurological events. When we analyzed the CAR-T infusion product, we found that as compared to the ZUMA-1 study, where the product was generated after 2 or more lines of chemotherapy, in this patients where they received only 2 cycles of frontline chemotherapy, we found that the product had a higher frequency of younger phenotype of CAR-T cells defined by CD45 or a positive CCR7 positive phenotype, suggesting that these T cells may have better fitness, and that in fact was associated with greater CAR T cell expansion that was observed on this study compared to the ZUMA-1 cohort. So in summary, I think this is the first study to evaluate CAR T cell therapy as part of frontline therapy for large B-cell lymphoma patients who have who met at least 2 high-risk criteria, one defined by histology or IPI, and the second by a dynamic risk assessment. And the therapy we found to be fairly well tolerated with a very high overall response rate as well as complete response rate of 74%, with 70% of the responses ongoing after a median follow-up of 9 months. We also found that the T cell fitness appeared to be better with the phenotype of the CAR T cells having a younger phenotype, naive phenotype, which was associated with greater CAR T cell expansion in these patients.
And what are some important takeaways from the study about axi-cel?
So I think as like any other new product, axi-cel was initially evaluated in relapsed refractory large B-cell lymphoma, where it was shown to be have very high efficacy. Subsequently, it was also evaluated in the second line setting on the ZUMA-7 study, which was a phase 3 randomized study, directly comparing axi-cel therapy with autologous stem cell transplantation. And that trial has completed enrollment, and the results are awaited. And this is the first time we are evaluating as part of first-line therapy. Now, this is an interim analysis of this product of this study. So longer follow-up and complete final analysis is needed to see if these responses are durable. But if if the long-term durability of the responses is confirmed, this provides a great need for this patient population who have a poor risk. However, it needs to be confirmed in a much larger study to be moved towards standard of care therapy in the future.
And as you mentioned, the study assessed this axi-cel in the first line setting. What would the impact be of a potentially approved CAR T-cell in the frontline setting for this patient population with large B-cell lymphoma if this were approved by the FDA?
Yeah, so I think we are still quite far away from approval. However, this therapy, currently patients with large B-cell lymphoma are treated with chemotherapy for 6 months or more. However, if CAR T-cell therapy was approved in the future, this could be significantly shortened the duration of treatment for these patients. The main advantage for CAR T-cell therapy is it's a single infusion therapy. And so far, the data looks like this therapy can be much more effective than standard frontline chemotherapy. So there's a potential to both improve the efficacy and curability of these high-risk patients. In addition, there's a potential to significantly shorten the duration of therapy for these patients.